with David Joseph from FLOGEN Technology. Please.
Thank you very much for having me today. I will present to you the work I have accomplished so far as director of the Biochemistry Department of FLOGEN Tech. This was my first paper, the fundamental neurobiological mechanism of oxidative stress related 4B2 protein deamidation. The paper has crossed 10,000 accesses in over 6 months as a single author paper. This was deemed by many media outlets as a significant achievement. The second paper has crossed up to 7,500, which was also deemed a significant achievement. Before we continue, I would like to make a quick shout out to my mother. She passed away before I completed my graduate studies. She was a famous lawyer in Canada. I love her very much, and I always will. I know we are mainly scientists, but in the field of molecular sciences, but my field is narrow. So, for the sake of a proper understanding of the work I did, I will start from the basics and follow up with the core aspects of my field. So, you have what we all study, DNA, which is the fundamental code of the human body, containing the genetic information necessary for the development of the cells of all living organisms. You have mRNA, which carries genetic information. The genetic information is taken from DNA and copied, transcribed into messenger mRNA. Nothing new here. We're all experts in this field. You have my subfield, which starts here. You have the mRNA 5' cap, which protects the mRNA from degradation. The mRNA 5' cap, like I said, defends from degradation. It is made up of a 7-methylguanosine, a nucleotide which contains no genetic information, but is essential for fundamental mRNA functions. A nucleotide molecule containing genetic information, the first of its line, and a triphosphate bridge that links them both. You have this very interesting protein, EIF4E, the eukaryotic initiation factor 4E, which directs ribosomes to the 5' mRNA cap structure. The mRNA 5' cap structure is connected by intramolecular forces to the eukaryotic initiation factor 4E, discovered by Dr. Witold Filipovich in 1976, a great scientist. It directs ribosomes to the 5' mRNA structure. And the ribosomes, as we all know, nothing major to see here, they're the macromolecular machines that copy genetic information from mRNA. Ribosomes use this genetic information to produce proteins, a well-known process known as translation. And finally, we have our proteins, which produce the chemical reactions which allow us to live. Proteins are the complex molecules such as enzymes, hormones, antibodies responsible for all the functions and structures of the cell and the living organism. Proteins produce the chemical reactions which allow us to live. Now you have this very interesting family of proteins right here, which are the 4-EBPs. 4-EBPs stop protein production. This whole line that you see here is hindered by 4-EBPs, for good reason. 4-EBP proteins, discovered by Tay and Lin et al. In the John Lawrence lab in 1994, attach to EIF4E, a process known as binding. This binding stops the recruitment of ribosomes by EIF4E. As a result, proteins can no longer be produced. So you have two competing factors here in translational control. You have EIF4E, which binds to the mRNA 5' cap structure and directs ribosomes for protein production. And you have 4-EBPs, which bind to EIF4E to stop EIF4E from producing proteins. You need a proper balance of both in order for these two competing factors to give you the ideal amount of protein production, of translation for the proper functioning of cells. 4-EBP2 is my protein of interest. 4-EBP2 is a member of the 4-EBP family found in the brain at a much higher rate than its paralogs, 4-EBP1 and 4-EBP3. 4-EBP2 controls all brain activity. It is responsible for the synthesis of the vast majority of proteins in neurons and in microglia. All chemical reactions in the brain depend on the proper functioning of 4-EBP2. 4-EBP2 is found everywhere in the body, mainly in the brain, but in all other organs. In the brain specifically, 4-EBP2 undergoes a chemical reaction called deamidation. Deamidation is a process that happens only in neurons, and this was discovered by Dr. Biochemistry et al. In 2007 and published in 2010 in Cell Reports. And this, what it does essentially is it blocks proper 4-EBP2 function, standard 4-EBP2 function. 4-EBP2, when deamidated, loses an asparagine, which is converted into aspartate, and can no longer properly bind to EIF4E. This increases protein production, which led to my research focus. On September 2023, I realized that a specific question had plagued the scientific community of biochemistry for 16 years. What causes neuron-specific 4-EBP2 deamidation in brain cells? This question was of fundamental importance for me because, like I said, all protein production depends on the proper functioning of 4-EBP2 in the brain. The discovery was as follows. On September 13, 2023, when I was 23 years old, I managed to discover the fundamental cause of neuron-specific 4-EBP2 deamidation. I will show you the process that I followed. The hypothesis was simple. 4-EBP2 deamidation happens only in neurons because of the axons and their unique properties. What are axons? They are long extensions of the neurons which conduct electrical impulses that are crucial to nervous system activity. I believed that the cables which link brain cells with each other are fundamentally responsible for this specific reaction, because axons are what make neurons unique relatively to other cells. I needed a specific range of a specific type of experiment to discover what causes neuron-specific 4-EBP2 deamidation, so I conducted what we all know as Western blots. 4-EBP2 in the brain is special. It appears as 3 distinct bands in the blot. You have the top band, which represents complete deamidation. You have the middle band, which represents partial deamidation. And you have the bottom band, which represents no deamidation. I saw this, and I managed to do a deamidation ratio, which has currently been coined as the Joseph ratio, to calculate the specific amount of deamidation that occurs in a part of the brain. You take the completely deamidated band at the top, you divide it by the band intensity of the band at the bottom. Mouse models were conducted for my experiments. You have 2-month-old wild-type mice, which are used as models because their nervous systems are representative of human nervous systems. The experiments were first conducted on the central nervous system. The experimentation was as follows. The first goal of the first experiment was to compare 4-ABP2 deamidation in the two parts of the neurons of the brain, the axons and the cell bodies. The optic nerve and the retina were chosen because they are a large extension of the diencephalon. So I needed to know if 4-ABP2 deamidation was happening specifically in axons because of axons. The problem is it's very difficult to isolate axons from cell bodies, as we all know, inside the brain because of the complexity of the organ. So I looked at the optic nerve. The optic nerve and the retina, I'm sure some of you may know this, is actually an extension of the brain. So when we say eyes are the gateway to the soul, it's actually 100% scientifically correct. The retina, the optic nerve are extensions of your encephalon, your encephale, as we say in French. And I extracted and dissected the retina, which contains mainly the cell bodies of the organ, of the optic nerve, from the optic nerve, which contains purely the axons when it comes to the neurons. And after this isolation, which was never done before for this specific type of protein, I obtained specific results which I wanted to compare with the brain. So I took pure axons from the optic nerve, and I took pure cell bodies from the retina. And I took the whole brain as well, which is a mixture of axons and cell bodies. You have your mixture, you have pure axons, and you have pure cell bodies, or soma, as we say in Greek, which gave the following results. So, deamidation, as you can see in this graph, is highest in the optic nerve and the brain. The brain has a lot of deamidation, 4-ABP2 deamidation. And when I compared the rate of deamidation in pure axons compared to the rate of deamidation in the brain, there was no significant difference between deamidation rates. But when I compared retinal ganglia, cell body deamidation compared to the brain and to pure axons, there was a massive difference. Optic nerves, pure axons had significantly higher deamidation than the retinal ganglia by a factor of 0.05 p-value. Retinal ganglia compared to the brain was even higher by a factor of 0.01 p-value. So no difference in deamidation compared to the axons and the brain, but a very significant level of difference in deamidation compared to pure cell bodies and the brain. The conclusion I drew from this, deamidation in the central nervous system is coming from the axons. You see the same thing when you compare the brain, which is a mixture of axons and cell bodies, to the axons, but when you look at pure axon, pure cell bodies compared to the brain, you barely see any deamidation. I wanted to extrapolate and see if this was not an isolated incident, but something systemic across the whole body. So I looked at the sciatic nerve, which is the longest, contains the longest axons in the human body. I isolated their cell bodies found in the dorsal root ganglia, and I also isolated their axons in the sciatic nerve. And lo and behold, same results. Even more significant here. We'll get to that later. Sciatic nerve had, by a factor of 3 stars, 0.001 p-value, much higher deamidation than the cell bodies in the dorsal root ganglia. My conclusion, the deamidation that we see in neurons is coming from the axons of the sciatic nerve. Just like the deamidation that we see in the brain is coming from the axons of our brain cells, our neurons. So, deamidation is coming from the axons, but what about the different types of axons? You have myelinated axons and you have unmyelinated axons. Myelination is like a kind of shield, mostly found in the neurons of your central nervous system, your spine and your brain. So, I looked at deamidation rates in your central nervous system compared to the sciatic nerve. The peripheral nervous system has much less myelination than your brain. And how did this look? You had much higher deamidation in your brain compared to the sciatic nerve. Myelinated axons showed much higher deamidation than unmyelinated axons. So, this led to, my experimental results led to the formation of my theory. My experimental results brought me to 3 conclusions. Deamidation is happening comes from the axons in the central nervous system. Deamidation comes from the axons in the peripheral nervous system, and deamidation comes mainly from myelinated axons compared to unmyelinated axons. So what does this show me? The science behind my experimental conclusion was simple. We don't need to go into the minutiae of deamidation here. I just wanted to give you an organic chemistry overview of the reaction. When I talk about deamidation, I talk about the amino acid asparagine converting into aspartate. This process is spontaneous. You don't need an enzyme for this. You take asparagine, you dump it in the water, it turns into aspartate. But this depends specifically on the structure of the amino acid, of the peptide asparagine is found in, which we'll get to later. Deamidation is a reaction that takes time to occur. This is the key. It's time. Deamidation is a reaction which occurs, which is observed 18 days after birth in mice. When the mouse is embryonic, when the mouse is a newborn, you do not see deamidation. You only see deamidation after 18 days of birth. So roughly 28 days after axons and neurons are first formed. You do not see deamidation in other organs of the body. So, you have deamidation, which takes 18 days. You have deamidation, which happens in brain cells. Why does this reaction, which takes 18 days after birth and 28 days after neurons are formed, happen specifically in the brain? I looked at many papers and I found that sodium proteins have SOD1, superoxide dismutase proteins, have a much longer half-life in neurons, in axons specifically, compared to other cells. SOD1 lives for 4 days in kidney cells, but lives for over 1 year in other types of cells. I concluded that this reaction is happening specifically in neurons because 4-ABP2 has a much longer half-life in axons compared to other types of
type of cells, which is observed in other species like superoxide dismutase. So this reaction takes 28 days to occur. If your 40BP2 has a turnover rate of 4 days in a specific cell, you're not going to see deamidation. But if 40BP2 has a half-life, a turnover rate after 40 days, 50 days, you're going to see deamidation. And according to many examples from many different studies, the ability of a protein to have such a long and extended half-life is only possible in neurons, and specifically in the axons of neurons. So, we see that 40BP2 deamidation is happening in neurons and axons because axons extend the half-life of the protein. Why do neurons extend the half-life of this protein? Well, if you look at your red blood cells and you compare the half-life of red blood cells with other type of proteins, you're going to see that proteins inside red blood cells compared to other types of cells have significantly longer half-lives. Why is that? Red blood cells lack a UPS system. The 26S proteasome is found excessively lacking in proteins, in red blood cells. You have here, in this study right here, you have in your red blood cells, sorry I'm getting cut off, we're beyond time, I'll make it as fast as possible. Red blood cells have a lack of proteasome. The less proteasome you have, the longer the half-life of the protein will be. Axons are very poor in proteasome, and this is specific to this organelle. Therefore, the half-life of the protein is much longer than expected in the axon specifically, and this was shown in myelinated axons. Myelinated axons are even poorer in proteasome than unmyelinated axons because of the lack of protein turnover, because of the lack of sodium channels in myelinated axons. So, axons are excessively poor in proteasomes, and this increases 40BP2 deamidation, and this makes 40BP2 deamidation a neuron-specific reaction. This was also confirmed by Ritchie, who found that 40BP2 deamidation happens specifically in skeletal muscle cells, but not in heart cells. And what is specific about skeletal muscle cells? They have axons ingrained in them, whereas heart muscle cells do not. So this led to the following principle. Due to their proteasome-poor environment, neural projections, namely axons, increase the protein half-life, which becomes more significant than the deamidation half-life of asparagines. The biological clock is now observed. My contribution fundamentally changed our understanding of protein production by demonstrating that the axon is a regulator of protein production itself. The axon causes deamidation in the 40BP2 protein, and by doing so stops 40BP2 from inhibiting the protein system. We're out of time, so I'll go as fast as possible to the logical conclusion of all this here, and then we're done. 40BP2 deamidation is axon specific. What is important about this? 40BP2 deamidation, the axon specific process which controls protein production in axons, allows scientists to target axon specific degradation for the first time. The importance of this is that you have in this process, you have for the first time a target which allows medical scientists to specifically target axon degradation, which is the key to memory loss. Memory loss is found, axon degradation is at the root of memory loss. And this was exemplified in multiple different studies. Axon degradation can even be reversed. Neurons, when they die, they don't divide. But when axons can reverse. Why is this important? We are currently in the process of completing a study, of completing a medication which targets this target within this year. The value of it, as estimated by Forbes, is $935 billion. And the next publications will be on this medication. So this was covered in the Associated Press. This is me with Nobel Prize winner Greg Semenza. Unfortunately, we were out of time. We could have discussed this much longer. I had a nomination for the Nobel Prize by Harvey Alter for this, and I was congratulated by the Prime Minister. Okay, thank you very much. If you have any questions, I'll continue there.
Thank you. Unfortunately, there's no time for questions.
No time for questions.
And I would like just to say thank you to all the speakers of this session.